Kelly, what is a biological population?
I don't know.
So there seems to be some confusion about what is a population. Yeah. Can you help us understand it?
No. Well, a population is the same species in a single area. Look, it's a separate population.
In a different area.
It's a different area.
Ergo, it has different germs and different biological substances in its whatever it is that require things to happen for life.
DNA is the word you're looking for.
Oh my gosh.
Yes.
That's so not, no, not dRNA,
Wait,
but DNA.
deoxyribonucleic acid. That's right. So
Oh my heck.
the thing that keeps our population separate is a dispersal barrier, seen here. This fence
This deck.
and the deck.
Elevation has a part to play also.
Yes. Now all these things combine to keep a barrier between these two populations.
And hence, with that barrier said, we are both going to be free of most diseases that are not prohibitively airborne.
Yeah, more than that though, we're not spreading genes between each other because we are not interbreeding.
Nope.
No.
Your population and our population are not interbreeding.
Are we a population?
Are you the same species?
Yes.
Are you exchanging genes?
Oh, that's embarrassing.
So, population ecology, we're looking at populations, a group of the same species in the same area that is exchanging genes. Okay? And we're looking at stuff like how big is a population, the life history, the demographics of that population, birth and death rate, population size, that kind of stuff. Okay. You also look at how a population is distributed in a given area, right? So are they clumped? Is it uniform or random distribution? These kind of things are determined by the environment and by the gene structure of a given population. So here's an example of a population genetics project that I worked on. I started out here in the mountains of northwest Arkansas looking for this little ball on a stick. It's an amoeba that forms a little fruiting body. It's one of these slime molds. Just happens to be the most boring looking slime mold of all. But I was trying to determine population structure in these things to see how far they were being distributed across the environment. Were they exchanging genes with each other across large scales? Okay. So I started out and I took a pinch of soil and dirt stuff, dead leaves, and then I went 1 centimeter away eastward and 1 centimeter away southward. And I went 10 centimeters, then 100 centimeters, then 1,000, and so on until I'd crossed quite a bit of the state on a logarithmic scale. Okay, and I took these and I grew them up, and then I sequenced a couple genes there, the 18S small subunit, the ribosomal DNA, and the intertranscribed spacer number 1. Okay, and I was looking for SNPs, single nucleotide polymorphisms. So how different were they, at least in these two little marker genes? Okay, I've plotted distance here on the x-axis and the number of differences in these genes between these isolates that I found. Looking at this plot, what pattern do you see? Well, if there was dispersal limitation across the state of Arkansas and a little slime mold from here over the past 20 years had not interbred with one over here, we would see differences in this group that didn't turn up in this one, right? If there's interbreeding going on, it tends to kind of wash out all the changes, or everything looks very similar. Okay, you don't have time to evolve a bunch of differences. Okay, so in that case, I would expect a diagonal line going from the bottom left towards the top right, in essence. The further you get away from each other, the more differences there are. I did not see that pattern. So what's this tell me? Well, it looks to me, at least from these genes, that we're looking at a single large population of this slime mold, meaning that interbreeding is happening between all these groups, at least on a time scale that prevents us from seeing a signal here. Okay. You can do the same sort of things looking at modern humans. This is tracking specific Neanderthal genes. Remember we talked about Neanderthal genes being incorporated into modern human genomes for some human groups. Okay. This is looking at the specific genes that got into those groups, okay, and how they evolved since humans moving through Neanderthal territory on their migrations out of Africa. All right, we see a few distinct clusters. All right, so here we've got one type of mutation that's happening in dark green, light green, and red. Right, so all of these groups have Neanderthal genes, but their genes are distinguishable from each other. All right, and down here in New Zealand, it's that light green also. So we can track human migration this way. Okay. So here's the European stuff. Notice that here in North America, we don't have any good examples. We're looking at indigenous people for the most part. Okay. We do that because they haven't been interbreeding as much with other human groups, and the signal hasn't been washed out. All right. So where did people who made it to North and South America come from? Well, the archaeology makes it clear that it was Asia, but so does the type of Neanderthal gene mutations. Okay. Another population ecology thing we can look at is demographics. So this is in your book, survivorship curves. We've got type 1, type 2, type 3. Okay. Type 1, we've got very few big babies that tend to survive pretty well. Okay. This is survivorship, the number of survivors on a log scale, okay, for every 1,000. So humans follow this. We tend to have babies that live into old age and then start dying off quickly. Okay. Whereas type 3, you have a ton of babies, tend to be really tiny, low parental care. Tons of them die off in the first part of their life because they just land in the wrong spot or whatever if they're a seed. Okay. And then if you get established, then you tend to live on into old age. Alright, and type 2 is stuff like squirrels, ducks, where you're equally likely to get killed all the time because life is terrible for you. Alright. Here's this playing out in basically real time with human populations. Okay. These are all human tribal groups that tend to have low gene dispersal between them and other groups. We're looking at fertility rate on the y-axis, offspring size at age 5 in kilograms on the x-axis. Alright, and here's the trend that we see. The lower your fertility rate, the bigger your offspring. Well, this is pretty crazy. Okay. So, what's it mean? Well, look back at this curve. This type 1 survivorship curve tends to have big babies. That means they're taking up more resources. Okay, and if you give your baby more resources, it tends to survive more. Okay, here, the smaller the baby, the higher their fertility rate. In essence, you have to have more babies if they're smaller to get the same number of surviving offspring. Okay? If you think about it, think of like a tree. An oak tree puts out thousands or tens of thousands of acorns in a given year. Okay? Most of those acorns, what happens to them? They get eaten by squirrels, they get buried by squirrels and rot, or they just land on rocks, die in the sun. Okay? Very few of them make it into germination in a suitable spot and survive. Acorns are also very small. They can get away with making thousands of offspring because they're really small and they don't have to care for them. Okay? If you have really big offspring and devote a lot of attention to making sure that they survive, you can't have that many. So this is played out over evolutionary time, just in the past 20,000 years tops, okay, where we see that fertility rates go down as babies get bigger. Oops. Okay. Why did they pick these groups of humans? These are all remote tribes. Well, they did that because there's not much interbreeding between these and outside groups, and evolution has had a chance to actually work on this without getting washed out.